share with you information today about using Arabidopsis GO annotations and the GO annotation tools that are available at TAIR. There are other tools on the web for analyzing GO data. I will talk only about the ones that we have at TAIR or the ones that we link to directly, but I can try and help. We can try and help if you have questions about the others. So we'll start with a very short poll, go into what GO annotations are, and what you can do with them. Then we'll have another really short poll and go straight into the question and answer. So Lenora, if you could please launch the first poll. Thanks. So tell us a little bit about what your primary research organism is and how much you know about GO right now. So if you could please answer those two questions. We'll close the poll out in just a few seconds. Thank you. All right, so most of you working in Arabidopsis and all of you have at least heard of GO before. So working in Arabidopsis, we know that we are working in an organism that is used as a gold standard. It's used as a reference for other plant genomes. What you may not know is that over the course of a year, over 3,000 articles are added to TAIR, and then about 2/3 of those are linked to one or more genes by a curator. And so we review those associations between the papers and the genes, and that's what shows up on the list of references if you go on the individual locus pages. We also extract gene function, expression, or phenotype data from over 400 articles a year. We don't have the resources to go through all the articles that come out in a year. And so you can imagine there's a big backlog of information that we don't get to. We would like to, but that's where other sources come in, like we ask the community to help. And there's also some annotation effort going on at bigger databases like Uniprot, for instance. But if you look at all the different sources, there are about 5,000 experimentally supported annotations that are added to TAIR using controlled vocabularies every year. And I'll go into what those are a little bit more in the next few slides. So first of all, what is a controlled vocabulary? It is an established list of standardized terms that are used for indexing or describing objects. And in our case, we're describing gene products, protein coding genes, non-coding RNA genes. And these are not just a flat list of terms, but they are terms that have specific definitions and relationships to each other. At TAIR, we use two main sources for controlled vocabularies. We use the gene ontology, and these terms describe a gene product's molecular function. So for example, kinase activity, cellular component where you can find it in the cell, like the chloroplast, or the biological process that it's involved in, for example, seed development. And then we also use terms from the plant ontology to describe expression patterns. So we'll capture where a gene product is expressed in the particular plant structure, so maybe the hypocotyl, or during which developmental stage. So maybe it's expressed during the torpedo stage of embryo development. If you go on TAIR, you will see that there's a keyword search, and keyword is the same thing as a controlled vocabulary term. A little bit about terms and their relationships next. And this is a figure that we'll go back to during the presentation several times. The related terms are arranged from top to bottom, going from more general to more specific. So you can see that the root term in this case is biological process, the most general term. And as you go down each relationship or each step, you see that the terms become more specific. So here now we already have flower development, that's pretty specific, but it becomes even more so when you can see the different floral organ development terms, carpel, petal, sepal, and stamen as the most specific terms. There are two relationships that are illustrated here. There's the ISA, so a carpal development is a kind of floral organ development. And a floral organ development is part of flower development because all the different floral organs together, the development of all those individual floral organs together constitute flower development. So that's what I want you to remember about this graph at this stage of the talk. Next, I want to give you a little bit of information on how annotations are made, and these are the links between the genes and the terms. There are computational approaches, and that's what's shown on the left side here. So it's the use of mappings and algorithms to take the sequences of the proteins or the sequences of the non-coding RNAs and determine certain domains or certain structural features or sequence features that allow you to assign a function. And so it may be that a particular protein domain is found in your protein of interest, and that domain has been found in another organism sequence, gene sequence, and then that particular protein has a function that has been determined in the lab. So then kind of by similarity, your gene or your protein is predicted to have that same function. In TAIR, we update the electronic or computationally made predictions every 2 months. On the right side of this slide are the manual annotations or the manual efforts as done by TAIR curators, by the community, and by Uniprot curators. And we collect all of those source annotations in TAIR. And that means reading the literature and reading the individual pieces of experimental evidence and structuring them and putting them into the TAIR database. And we do that weekly. So every week on the weekend, there's new information that goes into TAIR and is displayed on the TAIR locus pages. For example, a locus page like this. So you can search in the upper right box, you can put in the name of the locus, and you'll end up at a page that looks somewhat like this. If you were at Lenore's talk a couple of weeks ago, she went into detail on the various sections of the locus detail page. We're only going to look at this one right now, which is the annotation section. So you see gene ontology annotations and plant ontology annotations if they exist. And in this view, everything is compressed so that you just see a list of all the terms that have been linked. And the list is both electronic or computational annotations and experimental annotations. So you might wonder, how do I know which is which? This is when you can click on the annotation detail link, which is outlined in red here. And for that, you're taken to another page that contains more information. I'm going to blow this up even more so that we can break it down into the component parts. So here's one line from an annotation detail page, and it contains all the parts of a GO annotation. First, there's the gene product. In this case, it's CYPK25. Then there's the GO term, in this case, cellular potassium ion homeostasis. And then there's the reason why these two things are linked together. There's an experiment and there's a reference. So the experiment in this case tells you that there was a physiological assay done on a mutant, and that experiment was described in this paper. And from the blue color of this text, you can see that this is a hyperlink. And if you clicked on that, you would go to the page that describes the publication. And then you can go to the actual journal article if you want to read the experiment in detail. Now we go back to the figure that we saw earlier where I was describing the terms and their relationships to each other. And now we're going to kind of focus on using various degrees of specificity for annotation. If I were reading a paper where a mutant phenotype was described, where the carpels were disorganized or misshapen in a particular mutant of a gene, I would then annotate that gene to be involved in carpel development because that's the phenotype that's described in the paper. If I was reading a different paper and the phenotype described there was that kind of the flower itself, actually the individual organs don't form at all, there's like just the floral defect, then I might annotate something higher up, like for example, flower development. Because of the way these terms are related to each other, it's possible to aggregate information. And by that I mean, if I now go and search and I try and retrieve information, and I want to give me all genes that are involved in flower development. If you do this in TAIR, you will get not only the genes that were annotated directly to this term, but you will also get the genes that were annotated to any of the children. And so that allows you to kind of group gene products together, regardless of the level of specificity that was used for assigning the Go terms based on the publications. But even though you get this group of genes based on the general term, you can always trace back to the most specific that was used, and you can trace back to the publication on which that information was based. All right, so now we've got a little background on how annotations are made and the structure of the ontology. Let's go into what you can do with the annotations. So first of all, you can look at the individual gene level. You can look at the locus page and then see the annotations. You can also search for genes that are all annotated to the same term. So in this example, I have entered the term response to drought into my keyword term box. And that would request, give me all the genes that are annotated to response to drought or any of their children. There are many ways that you can restrict the search here, and to get to it, to get to this gene search, you would go to the to the search menu, and there's a drop down that would give you genes. But in this advanced search, one of the things that you can use for restricting is the evidence. And so you can pick, give me only the experimentally based annotations, and then you will get that list. So you can retrieve at the level of a single gene, you can retrieve at the level of a single Go term. You can also get annotations for lists of genes. And where might these lists come from? So you might have a gene family, you might have a set of upregulated genes in response to a particular stimulus from a microarray experiment, from an RNA-seq experiment. You might have a set of co-expressed genes that the things they have in common is maybe they're all in the same anatomical structure. And you want to see, tell me something about this group of genes. And you could use the Go annotations that we have in TAIR to do that, to see what they have in common. So for that, you would go to the Tools menu, down into Bulk Data Retrieval, and then there's something called Go Annotations. There are two buttons that you can use here. We'll discuss both of them, but I'll start with the Get to Go annotations. So you can upload a list of genes from your computer, or you can cut and paste a list of AGI identifiers into this box. If you click on Go Annotations, you will get back either an HTML table or a text table that you can download and then put into your favorite spreadsheet program. And that table of information will contain the name of the gene model. It'll contain the Go term that was used for annotation. And it will also contain a grouping term, a Go Slim term. And the difference between the two is that the glutathione transferase activity in this first example is the term that was used for specific annotation. That was the exact term for annotation. And then the Go Slim term transferase activity, this is a more general term which can be used for grouping. And we'll come back to Go Slims and grouping terms in a few slides. So this is going for groups of genes. You may also want to get annotations for the whole genome. So give me everything TAIR has for Go annotations. And for that, you would go to the download menu on the home page, go to Go and PO annotations, and then you'll see a set of files that gets updated on a regular basis. Two files of interest. One of them contains the word Go Slim in the name, and that has not just the name of the term that was used for annotation, but also the Go Slim term included. The other file that you may find interesting is the gene association.TAIR file. This is in the standard GAF format that the Go consortium uses for information exchange. So if you're doing something, if you're taking it to another tool that might use that standard format, this is the file that you want to get. And if you're interested in all the categories, all the Go Slim categories, for example, if you're wondering, what other things are there besides transferase activity and cytoplasm, then this file here, TAIR Go Slim categories, has a list of all those terms. All right. So now we're going to go into grouping genes by their annotations into broad functional categories, which means going to the second button on the tool that we were on where we got all the annotations. What happens if you put your list of genes into this and click this button? What you get is a grouping of your set of genes according to their annotations. And so there are three broad categories: cellular component, molecular function, biological process. We discussed this earlier. These are the three broad categories of gene ontology. And then for each of them, there are, I don't know, maybe 15, 20, 30, depending on the category, broader GoSlim functional categories. So there's a name that you'll see here. And then you have two columns of counts. One is the annotation count and one is the gene count. And you'll notice that the gene count is either equal to or smaller than the number of annotations. And this is because one gene may be annotated to, let's take this example of chloroplast. One gene may have multiple annotations to chloroplast based on different experiments, based on different papers, or based on different levels of granularity. So annotations and genes that are grouped in this category would be a gene that's annotated to chloroplast. If that same gene is also annotated to chloroplast stroma, it would be not counted again in the gene count, right? Because you've counted already once, but you would have one more annotation in this column. Here's an example of developmental process, and we're going to go back to our figure from earlier. So it's the same one. So developmental process will collect all of the genes and annotations made to this term and to any of the children terms. I hope that's clear. From this table, you can either draw an annotation pie chart, which is shown here, or a gene bar graph, which I'm not illustrating. Now you might wonder, I've uploaded my list of 200 some genes, and now they've been categorized into various categories. Does it mean anything if I submitted a list of 300 genes and 110 of them are in the functional category response to stress? Does that have any statistical significance? From this type of analysis, you can't tell that. So you need to do a statistical test called overrepresentation. And that's what I'll go into next. So finding functions that are overrepresented in your gene set. What we have at TAIR is a form that you can fill in with gene identifiers. And you can fill it in with identifiers not only from Arabidopsis, but from 9 other plant species that are listed here. What this then does is it takes the list of input genes and it sends it across the web to another website called PantherDB. PantherDB is one of the projects involved in the Gene Ontology Consortium, and they have developed the term enrichment tool or the overrepresentation tool for the Gene Ontology Consortium, and that's what this tool feeds into. In this example, I've taken a bunch of soybean identifiers and then selected soybean as the organism. So when you fill this in on the TAIR site and you click the button, it'll take you over to the Panther site. And here's an example of what the page will look like when you get to Panther. It'll tell you that you're running the overrepresentation test, and it'll tell you the annotation version and release date of the data that you are running your analysis against. And this is very important because you know right away how old the annotations are, how old the ontology is. In this case, when I took the screenshot, it was the March release of this year of the gene ontology, but they release every month. If you've been using other tools for Go analysis on the web, make sure you check how old the data is that you're running your analysis against. It can be difficult to find. So we like sending people to this tool, and we implemented sending from TAIR to this tool because we are sure that it's always using the most recent version of the gene ontology and of the gene ontology annotations. So then you've got the list of genes that you uploaded. the reference list, which is what is the background genome that it's comparing to. And then you pick, are you going to do an overrepresentation test on process, function, or component. Down here is some information on ID mapping. It tells you how many IDs were mapped, how many were not mapped, and then you go on and do the analysis. Here you have the results, and the view of the ontology is flipped from what we were looking at earlier, which means that the more general term is at the bottom, responsive stress is more general, and the more specific term, this positive regulation of transcription, is more specific and is at the top. So let's read the table and what does it mean. For the first line, it says that in the reference genome set, 51 genes were annotated to this term. The next is in the set that I uploaded, 4 genes were annotated to this term. The program has calculated that if association between a gene and a term is random, we expect in the number of genes that I uploaded as my test set, we expect to see 0.02 genes. However, let me back up and just do the math for you really fast. So there are 51 genes in the entire genome, and there are 55,000 some total genes in the reference set, so that's 0.09%. So in the set that I uploaded, there were 22 genes. 0.09% of that is 0.02 genes. So since we were expecting 0.02 and we got 4, that represents a fold enrichment of over 100. And then there's a p-value associated with that enrichment. So that's how you would interpret a result from an overrepresentation analysis that you've done through Panther. That is the end of my slides that I have for now.